Good day, children. Today I am going to teach you the next lesson of the series, which we are going to study under the coordination in Unit 5, Grade 13 Biology. Earlier we studied number of lessons related to coordination. Among those, the first one we studied Introduction to Coordination. Then we studied the structure and function of the central nervous system and the peripheral nervous system. And then as third lesson, I taught you generation and transmission of nerve impulses. Today is the fourth lesson, that is the sensory structures and their functions. So, as we know, we and the other organisms, all the organisms on earth live in external environment. So, they have to react according to the stimuli which actually reach from the external environment. At the same time, the multicellular organisms and the bigger organisms such as human has internal environment in addition to the external environment. So, there are so many changes taken place outside the environment. So, the temperature may be changed, the light, then we can say pressure changes. So, in addition to those, there may be lots of different changes taken place in the environment, not only the external but also internal. So, those changes which are known as the stimuli. So, these has to be detected from the body by certain mechanism. So, that is why we have receptors. Not only the human, the other organisms also have receptors which are important for detecting the changes in the environment. You know, the changes of the environment needs to be detected in order to survive in the nature. That is important for not only for the small organisms, not only for the all the organisms, it is very important. So, that is why receptors are developed. So, there are different kinds of receptors in organisms and animals. So, today we are going to study sensory receptors on humans. Human sensory structures and functions, that is the lesson. Before we move into the actual lesson, I would like to explain you the meaning of a sensory receptor. Sensory receptor, as I explained earlier, are specialized structures or cells or organs present in organism's body. So, they are to detect the external changes in the external environment as well as the internal environment. Specialized structure which can detect a specific stimulus. Second point, so it can convert the stimulus energy to a change in membrane potential to be transmitted. So, the light is taken as the stimulus and it has to be converted into another form of energy, that is the ability of the sensory receptor. Third one, it has to be connected with the central nervous system and it should produce action potentials to transmit into the central nervous system to obtain the perception and the interpretation. So, that is how we explain a sensory receptor. So, So, this is here. Now, you can see some sensory receptors found in human. Some are organs. As you see, the eyes, ears, nose, tongue, skin, all these are sensory receptors. Some may be structures like these, specialized cells, sometimes neurons acting as sensory receptors, specialized neurons. Next, what I am going to explain you is the basic characteristics of sensory receptors. So, those I explained already, some of them as a specialized structure designed to receive a specific stimulus, detect the stimulus if the stimulus is at or above the threshold level, sensory transduction, that is the conversion of the one form of energy into membrane potential, and connected, always connected with the nervous system, and has the ability to amplification and adaptation. Some of the words I want you to explain little further. So, this is also some of the specialized cells. Sensory transduction is the conversion of the one form of energy in the stimulus into another form of energy. So, that is how we explain the sensory transduction. Convert the energy of the stimulus into a change in membrane potential to be later transmitted as an action potential. Next one, amplification, another word that you need to know, is the strengthening of the stimulus energy by cells in sensory pathway. That is also important. If sensory adaptation is a decrease in responsiveness to continued stimulation into certain kind of smell for a longer period of time, so that person did not get the ability to get the changes of these particular the smell as a result of the sensory adaptation. Then the perception is the brain's construction of the stimuli. When these stimuli moved into the brain as an action potential, it has to be converted into certain form of the interpretation. It should give as a certain as a taste or as a object or something like that. That is done. So, the brain distinguishes stimuli from different receptors by the area in the brain where the action potentials arrive. So, that is done by the brain, that is we call perception. Then we move to the next one, types of sensory receptors. There are so many different types of sensory receptors in human body. Not only human body, other organisms also have different types of sensory receptors. These sensory receptors actually get the stimuli from different types of different changes in the environment. So, changes according to the changes of the environment. So, they are categorized into number of different types. First one, chemoreceptors. They can actually detect the changes of the chemicals which actually reach into the specific receptor. Thermoreceptors to detect the temperature changes. Photoreceptors to detect the changes of the light. Mechanoreceptors to changes of the physical deformation. Pain receptors, if there are any kind of tissue injuries or the sort of the wounds or something like that, pain receptors are to detect the tissue injury and so on. So, we need to study more about the detailed structures related to the chemoreceptors. The human can detect the smell and also the taste. Chemoreceptors can do olfaction, that is we call the obtaining the smell, and gustation, that is to get the taste. So, the both depend on chemoreceptors. Smell is the detection of odorants carried in air. So, that is you need to understand odorants carried in air and testants present in solutions. These are in dissolved form in a solution. So, olfactory receptors for detection of smell. Now, I told you two words, odorant and the testants. So, odorants, airborne molecules which are capable of stimulating smell receptors. Testants are water-soluble chemicals that produces a taste sensation by activating taste receptor cells. Now, in addition to these two, odorants and the testants, there are some chemicals which can be seen in circulating fluids, such as in blood. So, if you remember earlier lesson, we studied about the chemoreceptors which are involved in respiration. Some of the chemoreceptors are present in hypothalamus and some are present in the aortic body. Now, here you can see, this is here, you can see this particular structure. This is the part of the aorta. So, here you can see certain structures, these two structures which are called aortic bodies, aortic bodies. And another important structure which can be seen in this particular place in this carotid artery. So, you can see another structure here which has carotid body. These two are another chemoreceptors which are found within the body, which detect the carbon dioxide concentration, within the blood. So, we call it P carbon dioxide. That is also important chemoreceptor found in the human body. So, we are moving into the taste receptors which are found mainly on the surface of the tongue. Tongue is lined by stratified squamous epithelium. So, you can see the structure, it is a muscular structure which contain skeletal muscles lined by stratified squamous epithelium. So, although you see the surface of the tongue as the smooth surface, it is not a smooth surface. It contains this type of grooves. Can you understand? So, it forms a structure called papillae. There are large number of papillae on the surface of the tongue which contain taste buds insides. Taste buds. Taste buds are embedded structures in these papillae. So, it contains certain space here, you can see there is a space between papillae and the other area. So, that actually gives space for saliva to present. So, the saliva is important to dissolve substances or the chemicals which are present in the food. Taste buds get these dissolved substances into the certain cells present within the taste bud. Now, you can see here the taste bud is a cluster of cells, modified cells which are found within these papillae. So, these contain sensory cells. Now, you can see here sensory receptor cells. In addition to that, contain support cells. So, these sensory cells can identify different types of tastes. For example, sweet taste, salty, sour, bitter, and umami. So, here you can see, so if I moved into the next slide, you can see here taste receptors. It explains further, and there are 5 basic sensations of taste in a human: sweet, sour, salty, bitter, and umami. Umami is the taste which is actually obtained as a result of certain savory substances like glutamate, right. So, each type of cell here can detect different types of tastes. Now, this is, here you can see more detailed structure related to this particular taste bud. It is like an onion-like structure. So, the taste pore is present. There is a pore into which certain structures which are called taste hairs. Taste hair. So, as I explained earlier, there are actually 2 types of cells: taste cell, sensory taste cells, which are connected with, you can see the nerve endings, right. So, when the taste cell obtain the taste, so it forms, so when the taste cells obtain certain chemicals by diffusion from this region through taste hairs, so the membrane potential of taste cells change, which actually send into the brain through neurons which are connected with these. In addition to taste cells, there are supporting cells. Supporting cells that also important for taste buds to maintain its shape and also to protect the sensory taste cell. So, this is actually what I have explained so far is given here. Receptor cells for taste are modified epithelial cells. These are the things that you need to remember, very important. Modified epithelial cells organized into taste buds. This is how you need to remember this, right? Found in papillae. So, explained earlier, but I want you to highlight those things because they are important when you are answering the question. Papillae are small projections of the tongue, consist of taste cells, supporting cells, and sensory nerve endings. Those are the things that you need to write when you are answer the question, like if you are asked to write the short essay or something related to the taste receptors. taste sensation is done by different part of the tongue. This area for sweet taste, you can see, salty taste. So, this is the tip of the tongue, sweet taste. Salty taste, sides, more towards the tips. Sour, also laterally. Bitter taste in this region. So, this is how the certain receptor cells distributed on the surface of the tongue. Right. Pathway of taste. When a taste receptor is stimulated, the signal is passed to the sensory neuron. So, you have seen the sensory cells are connected with the sensory neurons. So, then each taste cell has only one type of receptor. Some cranial nerves pass signal to taste sensory area of the cerebral cortex. Then we move to the olfactory receptors. Olfactory receptors. So, this is for obtaining the smell. So, you can see it is found in the roof of the nasal cavity, roof of the nasal cavity. So, this yellow color structure, okay, which contains certain nerve endings which are moving down like this, okay, and it moves through certain bone. This bone is called ethmoid bone. So, you must so you may study it later in a later lesson. So, this area is formed from a special bone which is called cranial bone, which is called ethmoid bone. Ethmoid bone. It has this particular cribriform plate in which contains certain holes. Through these holes, sensory nerve endings moved into the upper surface of the nasal cavity, right. So, these are called olfactory nerves. So, when the person get the during the inhalation of air, the person gets the chemical substances which are coming from the outside environment. So, they stimulate these particular structures, right, and send impulses in brain. So, that is what here explained. Olfactory receptors are yellowish pigmented epithelium in the roof of nasal cavity near the septum. Olfactory receptors are specialized endings of afferent neurons, bipolar neurons, that convert olfactory stimuli into nerve impulses. Okay, right. Then, so it is receptor cells are here. It is actually a detailed diagram of the olfactory receptor. Detailed diagram. So, you can see this is the epithelium. This is the olfactory epithelium here. Epithelial cells are present in his, and among these cells you can see neurons, nerve endings, specialized nerve endings with cilia. Okay. So, this is actually present in a mucous membrane. This area contains mucus. So, the substances, chemicals, or we call odorants, which are moving through the nasal cavity, moving through the nostril into the nasal cavity, attaches with some of these odorant receptors present in the ends of the neurons, and changes the membrane potential. So, that is how the action potential is generated, and it is transmitted along the neurons present in the olfactory bulb through olfactory tract into the sensory region of the cerebral cortex. So, you can see in next diagram here, sense of smell occurs response to airborne molecules. This is the pathway of smell, nasal cavity, olfactory neurons, olfactory bulb, olfactory tracts, olfactory cortex in the brain. That is the pathway. So, this is another place these can be asked. So, to mention, you may ask to mention the pathway of smell sensation. So, you need to start from the nasal cavity. So, everything has to be written like this. Okay? Right. Next, we are moving to the mechanoreceptors. Mechanoreceptors sense physical deformation caused by the stimuli such as pressure, touch, stretch, motion and sound. These are the these cause physical deformation, right. Now, most of these mechanoreceptors are found in skin. Not only in skin, there are some other places also in the body, but we can find number of different types of mechanoreceptors in the skin. Among these, you can see here, it says Merkel disc, Merkel discs, or it says Merkel cells. So, which actually present very close to the epidermis. Then we have Meissner's corpuscles, Meissner's corpuscles. Then we have the Pacinian corpuscles, which actually found little deeper region of the skin. So, you just identify the places where you can see each of these sensory structures in the skin. Merkel's discs very close to the epidermis. Then the Meissner's corpuscles little below in the dermis. Then you can see Pacinian corpuscles little deeper region of the skin. And another one is here, free nerve endings. So, this is the diagram of the Merkel's discs. You can see here Merkel disc. So, it is actually found in the epidermis, more close to the epidermis, and it is connected with the neuron. This one, Meissner's corpuscle, it is actually a special nerve ending covered by certain structure, right. Then mechanoreceptors in the body include following: touch receptors. Now, I showed you Meissner's corpuscle and the position of the Meissner's corpuscles in the skin. So, those are touch receptors. They are not very deep, just the pass in something or the pass in the wind even you can feel, right. If someone touches you, you can feel because of the Meissner's corpuscles, sensitive to light pressure. Then you see Merkel discs, sensitive to light touch again, and free nerve endings are also for touch receptors. So, you need to remember all these. So, if you are asked to write the touch receptors in the skin, these are the three examples, right. Meissner's corpuscles, Merkel discs and free nerve endings. Pressure receptors. So, I told you certain kind of receptors are present deeper regions of the skin. So, that is to get the strong pressures Pacinian corpuscles. Example, which are present in deep skin. Some more. This is also a mechanoreceptor, vibration receptor. Vibration which are taken place in the external environment can be detected using these receptors in the body. Meissner's corpuscles, Pacinian corpuscles, right? Specific hair cells in the organ of Corti. Organ of Corti is found in inner ear. So, we haven't studied yet inner ear, right? Vestibule is also another important structure present within the inner ear. Semicircular canals are also important for vibration, to detect the vibrations. Okay? Sounds can be detected by these three receptors found in the inner ear. So, these are certain diagrams to show you, just to understand the positions of the vibration receptors in the inner ear. Organ of Corti is this area. Inside this cochlea contain organ of Corti. These are the sensory cells. Here you can see sensory cells. These are the receptors which detect the vibrations or the sounds, right, which is found inside the cochlea. Vibration receptors in semicircular canals. When we rotate in our head, or also when the, so that is actually the rotating, that changes are detected by certain structures found in the semicircular canals. Here you can see another important vibration receptor present within the inner ear, which is called the otoliths, so which are found in vestibule region. So, you may study these later in ear lesson. Then thermoreceptors. Thermoreceptors are to detect the temperature changes externally and also internally. External temperature changes are actually detected by the receptors found in the skin, right? Internal temperature changes, body core temperature changes can be detected by certain structures found in the hypothalamus. Okay? So, therefore, thermoreceptors are found in different part of the body. So, that is very important to regulate the temperature within the body also. Thermoreceptors carry out very important role in thermoregulation. So, therefore, we need to study about the thermoreceptors in detail, right. They are specialized temperature sensitive receptors which detect heat and cold on the body surface and in the internal environment of the body. As I showed you earlier, so you can see this particular structure which I haven't explained earlier, Raphini corpuscles. So, those are to detect the warmth or heat condition, heat changes. So, Raphini corpuscle is actually shown in this diagram also. In addition to that, free nerve endings also carry out certain important function related to detecting temperature changes. They are not specialized, they can detect the heat and also the cold. Body surface temperature is detected by number of different types of temperature thermoreceptors. One is called body end bulbs, or we call it Krauss end bulb, body end bulbs. In certain books it says Krauss and bulbs. Then that is to detect the cold. Raphini corpuscles detect warmth, and free nerve endings, as I explained earlier, both cold and warmth. Body core temperature is detected by special neurons found in hypothalamus. Then pain receptors. This is another type of receptors according to our categorization. Pain receptors are also specialized neurons, or we call them naked dendrites. So you can see here. These are found in the epidermal. They are extend to the epidermis. That is the important thing. They extend to the epidermis, the lowest regions of the epidermis, to detect these, to detect the pain. Humans, pain receptors or nociceptors are a class of naked dendrites in the epidermis. They respond to excess heat, pressure or chemical release from damaged or inflamed tissues. So, inflamed tissues or the damaged tissues produce prostaglandins, which actually stimulates the pain receptors. Okay? So, therefore, pain receptors are also important. These are very important for the survival of the organism, human, or mainly we are talking about the human sensory organs. Then photoreceptors. So, we are going to study in detail about the eye later, but before that I want you to show the photoreceptors in this particular slide. Photoreceptors are sensitive to light. So, human contain two main types of photoreceptors in the retina, in the eye, right, which are called rods and cones. Rods are like this, cones, they detect different types of light in the nature, right. So, some actually detect the darkness, black and white light during the black and white at night vision. Cones can detect the colors, right, they can detect the color vision. So, you can see here rods and cones are present within the retina, okay, rods and cones. Next we are going to study about very important sensory organ in human body, that is human eye. So, you can see here human eye, which is found in the skull region. So, present within the special bony cavities, right. It is actually round in shape. Two eyes are present in human. The eye is in the eye socket. So, it is a bony structure which is found in the facial region of the human. It is completely, it is covered, the eye socket is actually made from the bones. Some bones are actually the cranial bones, these are cranial bones. Some of these bones are facial bones, So, the facial bones and cranial bones forms a cavity in which eyes are present in human. So, they are very protected structures, so well protected inside these bony cavities. So, it is actually attached with the bones by, now if you take one side of one eye, in this side you can see the right eye of the person. So, the right side, so some of the face part is actually removed to show inside. So, then you can see the muscles, these are skeletal muscles which are attached to the surface of the eye and into the bone. So, these muscles are involved in rotating the eye within the bony socket. Now, these muscles are named as extrinsic muscles, which are required for convergence of the eye. So, that is to rotate the eye to focus into certain specific object. Now, I am moving into the basic structure of the human eye. So, the structure, as you see here, is made up of 3 concentric layers. 3 concentric layers like this. This is the outermost layer, so which is found around the eye like this. Outermost. This one is called outer fibrous layer. Outer fibrous layer. So, part of the fibrous layer here is transparent, that is found in the anterior region of the eye, that is called the cornea. But the posterior region and the lateral regions of the outer covering is opaque, and that part is known as sclera. So, therefore, you can see outer fibrous layer has 2 regions, anterior cornea and the posterior and lateral sclera. Anterior region is very important, that is the structure through which the light rays move through into the eye. Second layer is present just inner to this sclera. Second layer, we called it vascular layer, middle vascular layer, which is made up of 3 major regions. In this area, just beneath the sclera, it is called choroid, right? So, anterior region, you can see 2 areas are present. One is called ciliary body, and this ciliary body extended downwards, forming another area which is called iris. That is a circular region. If you observe in front of the eye, you can see circular region that is called iris. So, therefore, 3 areas are made from middle vascular layer, so which are named as choroid, ciliary body and the iris. Next one, Now, if I draw this, so you can see, so here, this is here. Like this. So, this part is the ciliary, this part is the ciliary body. This is what we called iris. Then behind this, there is another layer which is called the innermost layer. Innermost layer. There is another layer. Innermost layer starting from this part, this area, and move back. So, this layer is known as the nervous layer. So, which is having the retina. Right. This is the three basic layers found within the eye. Then we can see certain other structures inside the eye. So, we can see this particular structure, which is the lens, and there are two chambers in front of the lens and behind the lens. The one which is actually present in front of the lens is called anterior chamber. The one behind this is called vitreous body. Anterior chamber is filled with a liquid, clear, watery liquid. Vitreous body is composed of jelly-like transparent substance that is called vitreous humor. So, all these are present within the eyeball. All these are present within the eyeball. Lens, aqueous humor, and this area is having anterior chamber is filled with aqueous humor. Posterior, this chamber is filled with vitreous humor. Vitreous humor. Vitreous humor. So, those are present within the eyeball. So, in addition to those, so you can see some other structures. So, starting from the ciliary body, there are certain strands, right, which hold the lens in the position, which are called suspensory ligaments. So, they can be moved towards the lens or away from the lens, and the tightness of the these fibers may be changed according to that. So, in addition to this, you can see there is a depression behind the lens, right. This depression is known as the fovea, or we call it fovea centralis, which contain a special photoreceptor cells. And here, back of the eye, posterior end of the eye, you can see this structure that is called optic nerve, which contains sensory neurons to transmit impulses from the photoreceptors to the region of the brain where the perception and the interpretation occurs. So, here you can see aqueous humor and vitreous humor, and the lens, eyeball, the optic nerve. So, you can see the optic nerve. in front of the eye, there is another membrane. This membrane is known as conjunctiva. Conjunctiva. So, these are also important to protect the eye. Fine transparent membrane that lines the iris and front of the eyeball, this is called conjunctiva. So, this is to give you better understanding about this particular structure. So, all the structures are clearly shown in this particular picture. So, choroid, retina in yellow color, the transparent vitreous humor. So, everything is actually given. In addition to that, you can see this particular blue and red color tubes, which are the central artery and veins to the retina, which supply blood into the choroid. Choroid contain large amount of blood vessels, which actually provide the necessary required oxygen and nutrient for the retina. Basic structure of the human eye, sclera is white and opaque. It is the outermost layer of the posterior and lateral aspects of the eyeball, connects anteriorly with the cornea. So, sclera maintains the shape of the eye and gives attachment of the extrinsic muscles of the eyeball. Then cornea, so this is I showed you earlier, but you can actually see clear diagram here. Anterior chamber, this is the cornea, okay. It is made up of epithelial cells, and behind that you can see the lens. Also, cornea is devoid of blood vessels, doesn't have any blood supply, no blood supply, right, and involved in refracting light rays to focus the on the light on the retina. So, this is how you can see the cornea. Associated structures are also shown. Cornea, the anterior chamber with aqueous humor, lens. So, you can see that these are the two sides are having the iris. So, the iris is here. So, it is a circular structure, circular diaphragm with a small hole within it. So, choroid, ciliary body and iris you can see. This is how you can see the anterior side of the eye when you remove the cornea, right. Choroid, this is the ciliary muscle. So, these are the, this is here you can see, this one is the iris. Iris is having a small hole within it. So, that actually controls the amount of light entering into the eye. So, it can be expanded or dilated or constricted. Pupil can be dilated or constricted according to the nervous supply by autonomic nervous system. So, the choroid iris contain two arrangement of smooth muscles, circular and radiating. You can see circulating muscles and radiating muscles. So, the contraction and relaxation of these muscles involve in changing the size of the pupil. in addition to the I explained most very important area about the receptors today. I taught you chemoreceptors, then thermoreceptors, mechanoreceptors, and the photoreceptors also. I have to continue the photoreceptors further, that I will do in next lesson. Until then, goodbye.